And now I'm going to share my screen with our doc cam here, okay? So here we go. There, perfect. So you can see here's my lab setup. Okay, everyone, if you're following along at your desk, you can flip your test tubes over so that you can actually work with them now. Okay, so you've got just our 2 test tubes. Okay, now if you're at your desk, you should have a small beaker with yeast in it. Okay, and if you don't remember, this is what yeast looks like. We see the tiny grains. Okay, they may not look alive, and maybe we would describe these as having the potential for life. Given the right circumstances, they will start to metabolize and become active. Okay, so what you want to do first is we're going to make our yeast mixture. And so literally, you are just adding the yeast to the water. So you can pour your yeast into your water, not in the mixture. That would mess it up. So Braden made a good point. Do you add
I don't
sugar
think it's
too?
sharing yet.
No, right. So you do
Okay.
not share, or I'm sorry, you do not add sugar to this original mixture. You're going to add sugar to one of the test tubes, but not the mixture. Yep. So just pour it in. Yep. Then take your spoon and you're going to stir it. Okay. You want to make sure there are no clumps in the mixture. There are no clumps in the mixture. You've just got yeast and water. So you have a lot of water there. Okay. I would make sure you try to get all that yeast out of there so that it actually is all mixed together. You don't want to see any clumps, so stir it for about a minute. Okay. Now, if you are following up here, okay, so I've added the yeast to my water. Yeah, this can get messy quickly, so just try to be. And it is hard to break up the clumps, so like I'm doing on here, I'm just mixing this together, try to break up the clumps, and then just keep stirring. And you have kind of like a soupy mixture. Okay. Now, as Braden pointed out, okay, what is the or what are the ingredients to this mixture? What are the ingredients to this mixture? Angelina? Excellent. Water and yeast. Okay. So you can see now, look, the clumps are gone. Okay. I just was stirring it. The clumps are gone. Okay. And so this is my water and yeast mixture. Now you'll notice on in the containers that I gave you, there's a little lip here, this little lip. That's to help you pour this mixture out, okay? So what you're going to do next is, oh, hang on. You're going to take your mixture, okay? I would actually take your test tube out of the rack, and you just want to fill this test tube about a third of the way up, You're not going to use the whole mixture. You're putting it in both. Okay? So about a third of the way up, just like that. Can everyone see what I've done there? You're filling it about a third of the way up. Okay? And now do that in both test tubes. Try to make them as equal as possible in terms of where you fill it up to. Oop. Perfect. Okay, good. Mine is not, so I'm going to dump a little back out. Perfect. Okay, so now I have 2 test tubes. Now, as of right now, these test tubes are identical. They just have a yeast and a water mixture in them. Anyone need help? Perfect. Setup so far looks good. Okay. Good. Okay. Good. Okay. So we're in the home stretch now. What we do next is you're going to need your balloons and your sugar. So before you touch the sugar, hang on, sorry. Stretch out the balloon, one balloon, doesn't matter which. Try to use 2 different colors, that'll help you keep them separate. Test tube 1 is going to be our control. So that means we're not going to add anything to this yeast and water mixture. That means that this yeast and water mixture should not have a food source, okay? Nothing to metabolize, okay? So what you're going to do after you stretch your balloon out a little bit, put the balloon over the lip, okay? This can get a little difficult, so be patient and just let it sit, okay? That's number 1, okay? Now, according to what we've said, Amaya, what should I add to test tube number 2 if this is our experimental group? What do I need to add to this?
Sugar.
Yes. So you do need to add sugar to your second test tube. So what you'll do is take your sugar. Now you have a packet of sugar. I want you to add about 1/2 to 3/4 of the packet into your test tube, okay? So here I poured mine out already, so now I'm just gonna add the sugar in, okay? Oop, there we go. Now again, this is just table sugar, okay? Just sucrose. Oop. Okay, so I've added the sugar in. Now you don't want the sugar sitting on the bottom, right? So what you'll do is put your thumb over the top, everybody. Thumb over your top, shake it, okay? Make sure there's no sugar in the bottom of the tube, sitting on the bottom, okay? And then you want to keep your thumb over this and then try to cover it up as quickly as possible with the balloon, okay? Shake it first, just so there's no sugar sitting in the bottom, okay? So just to make sure it's mixed, the bottom should not have any sugar granules in the bottom, okay? So there we go. Now our experiment is set up. So you should have your data table up now. We are at minute 0, and now we're at the point of the lab where we can record our data. I would leave the gloves on if you can, okay? If they're too tight or too loose, I can give you a different size. Okay, so everyone has it set up? Okay, now if for your lab setup, you're gonna be recording your own results. If you're my lab partners today, you'll be recording the results that I record, okay? So everyone should have a ruler on your table. Now, how do you think we could measure, what do you think we could do to measure the carbon dioxide production here? Assuming that any gas that goes into the balloon is carbon dioxide, yes. How big the balloon gets, okay, so we could measure could we call that circumference of the balloon? Okay, so we can measure circumference. Or if you look right here, where the bubbles, where the surface, you may start to see bubbles form there, okay? The bubbles, of course, would be carbon dioxide, okay? And you could also measure there, okay? I'm gonna measure circumference, okay? Whichever you choose to measure, just make sure you stay consistent and you measure that each time, okay? So this is minute 0. Take your first set of measurements now, okay? Now, so my lab partners, what we're gonna do, millimeters, do millimeters, so the smaller side, okay? So, Lexi, if you look at my ruler here, can you see this? Okay, so this is the balloon I'm measuring right here, and I'm gonna try to measure the circumference. So, what does that look like to you?
Like the wait, which side am I looking at?
So right here, this is what I'm measuring, the circumference. So what does that look like to you?
I'm confused.
Yeah, so 0, right? It
Oh,
hasn't inflated at all yet. Exactly. Okay. So now, so for test tube 1, we're just putting 0 millimeters. Okay. Now, this is test tube 2. Okay. Okay. Also 0. Good. Okay. Then now that would make sense. Cell respiration doesn't happen in an instant. It takes a little time if we're going to see any gas build up there. Okay. Did anyone get anything different than 0 for their first set of measurements? You did? Okay. Really? Okay. So, all right, I'll come check that out. All right. So what? Yeah, exactly. Yeah, good job. All right. So if you are one of my lab partners right now, so if you're remote right now in the chat, what I want you to do, tell me the difference between tube 1 and tube 2. Okay. You can send it as a private message. You don't have to send it to everybody. Okay. Just send it to me. What's the difference between tube 1 and tube 2? Okay. Now I'm going to set a timer now, and we're going to do a series of 4 more measurements today. Okay. So let's do 4 minutes. Okay. So in 4 minutes, we'll take another set of measurements. Okay. And I'll put the timer right here next to our test tubes. Okay. I'm going to do a quick laugh and make sure that everyone's set up correctly and feeling comfortable. And then we're going to talk about what happened last night. Okay. All right. Sound good? 0, 0. Okay. What was this? Cool. Okay. Good. Okay. So are you doing the bubbles? Cool. That'll be interesting. Good. Good. Okay. 0, 0. 0, 0. What'd you get? That's not 0. No. Yeah. Now it's after the time though. So what I would do is just let it. What did you write down for your first measurement? Mm-hmm. So if each tick is 1, so 20 wouldn't be until up here. So I would do a little lower than that. Got it? 0, 0. Okay. Okay. Which one was? This is your sugar one. Got it. Okay. Are you doing the bubbles? Got it. Okay. Perfect. Okay. So we've got about 2 minutes to our next measurement. I'm going to check answers now. Let's see. Okay, tube 1 has nothing added to the yeast and water solution. Tube 2 is sugar added. Great, Maya. Good. So, Tim, what are we expecting the sugar to do for the yeast?
It will produce carbon dioxide.
Excellent. So what process is it undergoing?
What do you mean, what process?
So what process would produce the carbon dioxide?
Oh, cellular respiration.
Exactly, respiration. Okay, good. Cellular respiration. Now, I'm going to stop sharing momentarily, okay, so that I can show you some pictures from yesterday, okay? So just hang on 1 second. Here we go. Okay, so we only have about 40 seconds, so we're going to take our next set of measurements and then I'll show you test 4 from last night, okay? So we have that set up, ready to go. Okay. Everybody is comfortable with how you're measuring? Okay. So, Ella, what box are you going to write this next set of measurements in? What time are we on now? Good. So you'll write this next set of results under, should be under 5 minutes. Oh, so wait, this, you what? But there's no sugar in this one, right? Okay. So then you want to think about what might have caused that. Okay. All right. So take your second set of measurements. Take your second set of measurements. Okay. And then record them for 5 minutes. Okay. My lab partners, we're going to record our second set of data now. Okay. So we'll start with test tube 1. This is just with yeast and water. There's no sugar in here. Okay. So I'm trying to press down just to see if there's any gas in the balloon, and there's not. So I'm still going to just measure it just in case. Okay. And still, we're at 0 here. Okay. Now, I do see some bubbles here in my test tube, and I'm going to zoom in so you can see those. So if I go onto the side, I can see that there are some bubbles forming in here. Oops, sorry. There we go. So there are some bubbles forming in here, but the balloon has not really inflated very much. There's only a little bit of gas in here. Okay, so I did try to trap that in the top. Okay, it now did pinch out a little bit. So let's see. So that's right at 10, but not quite. So we'd say 9 millimeters. Okay, for beaker number 2. Okay, 9 millimeters. You've got 0 still for tube number 1, and 9 for tube number 2. Okay. I am going to ask you to send me a picture of the data table today, and then at the end, we'll have a record of our results. Okay. So just make sure you're keeping up with that. We've got the timer going for our next set of measurements. I'll move that in front of the clock for you. Okay. So here we go. Okay.
Mrs. Durante,
Yes, Tim.
do we have to include information about the bubble in the tube? The
You do
second
not.
tube?
No, I just wanted to show you that just because you can't handle them in person, I just wanted you to see that that was happening. Okay. So Tim was just asking guys about the bubbles that I showed in the tube. So hopefully you can at least see some bubbles, even if you're not getting much inflation yet. Okay. And again, bubbles would be a sign of carbon dioxide formation, right? And so no, you don't have to write that down, but I just wanted you to see it because you're not seeing the test tube right in front of you. Okay. So right now we're testing respiration with the yeast. Last night, so Amaya, Lexi, Peter, who else? Tim, you took supplies home, correct?
Yeah.
Did you get a chance to do the lab last night?
Yeah, I did.
Okay, so I'm actually going to disconnect for a moment. So when you speak now, instead of talking to me, you're going to be over the speaker in the room so everyone can hear your results. Okay. Okay, so do you want to talk to us about your results from last night?
Nothing happened for us.
So nothing happened?
No, the color pretty much stayed the same. I mean,
Yes.
Lexi a little bit, but nothing else happened.
Okay, so when you say a little bit, what do you mean?
It got lighter.
It did get lighter. Okay, Peter said
But not like drastically.
not drastically. Okay, and now let's remember what we did yesterday. So we started with cabbage juice that looked like this. It's an indicator for an acid, right? Okay. Now, I'm going to hold up here. I'm going to hold up the solutions. So would you say it turned this color?
No.
No, not that.
Not that?
No,
Okay.
just light purple.
Just light purple. Okay. So that tends to be what happens. Okay. So you're not going to see it go totally pink like we did, say, when we added vinegar, right? So the carbonic acid that's going to form, obviously that's a chemical reaction. So that takes time. And that's why you have to keep blowing for like 5 minutes, even to see just that little bit of change. Okay. So what you would want to say for test tube number 4 is it turned light purple. Okay. So it didn't go pink like the vinegar, but it did go light purple. So would that be evidence that you're exhaling carbon dioxide? Would that be evidence that you're exhaling carbon dioxide?
I mean, yeah.
Yeah.
Slightly.
Yeah, to some degree. Now, do we know how much?
No, not exactly.
No.
So it's an indirect measurement, right? So not perfect, but it gives us some idea of that process is happening. Okay. Now, if your balloons are starting to look like they have pockets of gas in them, okay, you can kind of try to straighten them out or push the gas pocket to the top. Okay. And it's time for your next measurement. So make sure you're doing that as you perform your measurement. Okay. So I hate to tell you guys, for my lab partners, but my results are not as good as, or not as clear as some other people's. Okay. But again, if you look, no pockets of gas in this first balloon. Okay. So again, I'm going to measure it to be sure. I'm going to zoom out again. I'll measure it to be sure, but it's not looking like anything has changed. Okay. And yes, I'm still at 0. Okay. Still at 0. Now let's move over. Oh, look it. So there is more of a pocket in here. It's just not as pronounced. Okay. So let's see if it's gone up at all. A little, but not much. So I'd say that's right at 14. So you'll have 0 and then 14 millimeters for your next box. Okay. You have what? You have more than that? You, it is, yes. So there's clearly gas in here, though. Which, if yours is not reacting, guys, look at the bottom and see if you have clumps of sugar in the bottom. Do you see like those grains of sugar? Oh, yeah. That might mean you need to mix it up again, just so the. Ooh, see? Okay. All right. I would probably mix that up again, but you want to cover the top because you don't want the liquid going into the balloon. Okay. Yeah. Did that change your results at all? Okay. Oh, look it. See? So at this point, guys, just so I can kind of describe to you what's happening in class, you should start, people are starting to notice a pretty large increase in the amount of gas being produced right now. Okay. Now, the next timer's already been started. Okay. So make sure you're recording your results. Okay. Amaya and Lexi, do you mind if I show the picture that you sent me?
Sure.
Is that okay?
Yeah.
I won't show your email, just the picture.
Okay.
Okay. So question. Braden just asked, what do you think would happen if you put the sugar in the yeast and water mixture that's sitting on your table? Yes. So what if you put the rest of the sugar into that mixture? Okay, so you would see bubbles, but you can't fit a balloon over the top, so you wouldn't necessarily be able to capture it, right? So where would the carbon dioxide go? Yeah, into the air. Okay. So same idea about like why does Coke or a carbonated beverage go flat? Okay. Over time the carbon dioxide diffuses into the air. Okay. So we'd have to come up with a different way to measure that if you wanted to do it. You're welcome to do that if you want. You might see some bubbles forming at the surface, but go ahead if you want to try, and you can see if anything different happens. Okay. Okay. So as we said here,
Mrs. Duranti, this is the
yes,
fermentation. We don't need oxygen, right?
correct, correct. Yes, that is correct. Yep. It does smell like bread, and why do you think that? That's what we use. Yeah. Okay. So wait. So Amaya, Alexi, where did you send me your results? I'm not seeing them.
I emailed them to
Who emailed them?
Alex.
Alexis?
Yeah.
Okay, so you got about 20 seconds, so get ready to measure again. Oh, you sent it to me on Schoology. That makes sense. Okay. Okay, take your next set of measurements. Okay. So next set of measurements. Okay, again, not any change on the blue balloon. So you can see I'm pinching it here, still at 0. Still at 0. Okay. I'm gonna flip this around to get so you can get a little better view. Uh-oh. Test tube tried to escape there. Okay. So now you're looking at the orange balloon. Okay. Again, I'll try to trap some of the gas in the top. Oop, oop, hang on. Okay. So again, I'm trying just to push the gas to the top so I can measure it. Okay. It looks like it has increased again. Oop. Try to switch my fingers around here. Okay. So now we are at 22, about 22 millimeters. Okay. Okay. And now that is starting to inflate. It's starting to stand up on its own. And if you look, I can still see bubbles at the base here. Okay. When I look at test tube number 1, no bubbles. So it shows me respiration is not occurring. Okay. And again, this is not standing up at all. No gas is being trapped up here. Okay. There we go. Now I already started the countdown to the last measurement. I believe you have 2 more spaces left, correct? We're gonna stop. We're gonna do one more, and then we're gonna stop, because then we do have to do a little bit of cleanup today. Okay. So if so, my lab partners, you can cross out that last box number 5. Okay. And then um, we'll go from there. Okay. You what? Yeah. That has happened. Okay. And I'll show you at the end what happens when you take the balloon off the top. Okay. There's actually a lot of pressure building up in there, depending on
Mrs. Duranti,
how it's going. Yes, Tim.
what was the result for 15 minutes of the second tube?
22. 22 millimeters.
20.
Yeah.
Thank you.
Yes, for just this last one, right?
Mr. Ranty,
Yes.
can I ask for the 10 minutes for the test tube too?
So what did you get for that one, Lexi?
I got 14.
Good.
For-
Yeah, exactly. Yep, that's correct. Okay. Oops. Okay. So this is what we'll do. I'll post the picture for number 4. I'll post the picture for number 4 as an update in Schoology today, okay? All right. Okay, so did everybody hear that? The picture for test tube number 4 from last night, I'll post those pictures as an update in Schoology today, okay? Now, so that should help out with test tube number 4, okay? Just light purple, according to most people. It just turned light purple, so it didn't go all the way pink like it did for the vinegar, okay? So some reaction happened, but maybe not what we predicted. Yeah, if that makes sense. Okay, so less than a minute, and then we'll take our last measurement, okay? Okay, and so you guys crossed out number 5, correct? Yes. So by number 5, I meant the 5th box. So you're not doing the 25 minutes, okay? Jason, is that blinking out on you guys?
No, I see it.
You see it now. Okay, great.
Mhm.
Okay.
Yeah, it was loading before, but I see it now.
Okay, got it. Okay, take your last measurement, guys. Take your last measurement. Okay, no surprise here. Test tube number 1 is still at 0. Still at 0. Okay. Oop. Still at 0. Okay. Now let's flip this around. So for the last one, so you can see gas is visible. Okay. Don't know if it's gone up very much, but let's check it out. Not much of an increase. So I would still say that's still at 22. 22 millimeters for tube number 2. So 0 and 22 again. Okay. All right. Now, for cleanup today, okay? And so now, if you are my lab partners for today, obviously I'm doing the cleanup for you, so you can start looking at the conclusion questions, okay? I'm not going to mute myself so you can hear kind of the overview here, but you can start looking at those conclusion questions. Okay, so for today, there are a couple things I need your help with, okay? No one else is using these lab supplies today, but I'm recycling. I don't want the spoons thrown away in particular, okay? So basically what you're gonna do for cleanup today, I'll show you with my supplies, okay? We'll get up one at a time. Actually, you can even probably do this at your table, okay? You're gonna take these balloons off, okay? And just make sure that they don't like rip on the tube, okay? We're gonna toss those out, okay? Uh, then you just wanna dump out the contents in the sink, and there's already a bucket in here with dish soap in it. Just put them in the dish soap, okay? Does that make sense? Uh, then any extra balloons, sugar wrappers, those things you can throw away. Everything else I'll clean and sanitize later, so you can just put those up here on this front table. Okay. Any questions on that? So I'll send you up a couple at a time. So Matt, you can start. Okay. So just put your test tubes in the sink and then bring me everything else. Okay. Leave your gloves on until you're done. But when you throw your balloon out, you can throw your gloves out too. Yeah, just leave them in there. I will take care of that. So you can just leave it on there. Okay. I will take care of that. Okay. So as people are cleaning up, just heads up for tomorrow. You leave it on your tray. So heads up for tomorrow. So at the start of class, okay, we're going to talk through the results for both of the labs from this week. Okay. Then keep in mind we're done with John. You can go now. We're done with cell energy. So we are reviewing Thursday, test Friday. Okay. Thursday in class, we will do the full review. Thursday night, I will have a review session. I said 5 to 6. If that's a problem for anyone, let me know. If it is, you should schedule time with me tomorrow. Okay. I know that's a lot of information, but that's photosynthesis and respiration. Okay. Yes. Mariana, you can go.
Mrs. Uranti?
So we didn't get a chance to go over number 4 in detail. I would wait, but if you are confident in your answer, you're more than welcome to. It's not going to be late though if you wait until Wednesday. Okay? Does that make sense? Just leave it right there. Yeah, for right now. You're good? Okay. All right. Any other questions? Okay.
Mrs. Uranti?
Kim, yes.
For question number 10, like, why do we need to have 2 test tube? Can I say, like, decide to compare and see the change between 2 tubes?
That would be exactly it. Yeah, guys. So Tim has a really good question. He said, why we didn't use 4 test tubes, but if we had, why would the original lab have been set up that way? Mariana? Exactly. Oh, and for consistency too, right? So tomorrow when we go over results, we're going to make averages of all of our data, and then that'll kind of give us an idea of where everybody was at. If there were any outliers, okay, or anything like that, okay? Well, so the idea is that it's always better to have more tests, more trials, right? For our purposes today, if we could have, it would have been nice to do more, but just in terms of materials and time, we couldn't, right? So to make up for that, you can go. To make up for that, what we're doing is everybody did a lot of tests individually. So instead of being in a group of 4, you're in a group of 1. So we still have a lot of data that we can look at tomorrow. Does that make sense? Would it still have been better if everybody had done 4? Yes. It's more reliable if that way, but right. You would have done 2 of the controls and 2 of the sugars. Yeah, just to see if there was any variation. Okay. All right, you can go put your tubes in the sink. Okay. John. I would wait until tomorrow because we do need to talk through the results a little bit, and I do want to make those averages with you first. Okay. Leave it all on that tray. Just the only thing you're putting in the sink is your test tube. Okay. Okay. So I will actually just come grab these for you. I think that'll work better. Okay. Done. Can you push that down? Did you just push your supplies to the end of the table, guys? I'll take care of cleanup on these today. Okay. If you're remote, you can sign off. I'm gonna clean desks in a moment here. Um, I would definitely recommend at least looking at that review tonight so you're ready for tomorrow. Okay. All right. Peter, When Sammy's done, you can go. Okay. Good. Good. Thank you. Thank you. Okay. You done, Sammy? Will you put your ruler on there for me? Thank you. Okay. You guys can go. I will spray your desks. Just do me a favor and don't leave until you've returned your supplies. And I will see you guys tomorrow. Oh, so just try not to crowd by the door, okay? Give yourself some space. Whose was this? Peter, you done? Okay, perfect. No, I just want to make sure. Ladies, you put your beakers where they belong? Okay. Good? It worked out okay? Good. So what, did they do anything at all? No? Okay, so let's take a look at that. When we have the averages tomorrow, that's what you want to compare it to see how different your results were from the averages. Does that make sense? Then we'll have a better idea of how off it was. Hey. Oh, cool. Okay. Okay. Perfect. Okay, perfect. Yes, sir. All right, I'll see you later. Hey. Okay, so give the dust another like 30 seconds, okay, before we wipe them off. So you can go sit down in your seat, but just the dust are not ready to be wiped off yet. How's everyone's morning? Good? Okay. So I'm gonna